great pleasure to be here today and to see you all here. For those who don't know me, I'm Danny Alexander. I'm the Director of the Centre for Neuroimage Computing here at UCL, and soon to be Head of the Department of Computer Science as well, both of the things that James works within. So I think, these are really one of my personal favourite events in the academic calendar, where we get to gather and sort of celebrate someone's achievement over the years and hear about their story. We're trying to keep the culture of doing these inaugural lectures as much as possible. So thanks, James, for stepping up, leading the way, as usual. So just a few words about James before we start. So James joined us, I think it was 2019, was it? Yeah, it's been here 5 years. I mean, it feels much shorter than that, actually. So, but I don't know what that says. But joined us in 2019 at the Centre for Neuroimage Computing, and actually joined us as a kind of partly at our centre, but also working within the Dementia Research Centre. It was really in a liaison role between the two centres, and it's been really magnificent in that role, because it's not an easy thing to do, I don't think, to be a bridge between two different centres, and to do that in a magnanimous way that not only kind of helps your own research but helps everybody else within both centres. But James has really done that in a really nice way, so I think we're all grateful to you for that. Has made loads of other contributions to both the centre and the department while he's been here. Just to pick a couple of things, he was part of the Equality and Diversity Committee for a long time, and that's something that James feels very passionately about and has really kept us in the centre informed on that and contributed a lot to the department, shaping the culture of the way that we work. He also led, recently, the acquisition of a new piece of kit for us, the high-field length-field MRI system. Again, typically magnanimous approach to that, and persistent approach. How many times did you put that proposal in?
7. Yeah.
So I lost count.
I hadn't, but
brilliant achievement in the end, and that's given us all a new dimension to the work we do here at Senior. And more generally, I always think of James as you're a real heart and soul person of the centre. You're always in having lunch with the students and the postdocs, and knowing everybody, and as a real social centrepiece for the centre and the department. And so we're all very proud to have you as part of the centre. And I'll shut up and make way for the good stuff. Over to you, James.
Yeah, thank you very much, Danny, for the kind words. Feels a bit surreal to be standing up here in front of you guys and talking, doing my inaugural lecture. But thanks for Danny for pushing me to do this. And I really enjoyed Ivana's event when we had that last year. I can't decide whether to wear the hat when I'm talking or not. It's actually really hot, so I might take it off and rest it here. And I'll make a start. So when I was trying to decide how to kind of frame this talk, I thought somehow I've got to work in some kind of pun relating to brain age, because that's been the main topic of my research over the last 10 years now. And I thought, actually, obviously, I like my ancient history and I like my classical culture. So I thought, I know, good old Ovid. I'll refer back to his Metamorphoses and talk about the 4 ages of man, but not all men or all humans, but 4 brain ages of my own career. And really, metamorphosis is a good word, because I kind of see my career as a series of transformations in many ways. But as well as transformations, I also want to say my career is one where I really need to show a lot of gratitude. So I've been really, really fortunate, basically throughout my whole life. I've had very, very supportive family, very supportive friends, and great colleagues everywhere I've been. And I definitely couldn't have got anywhere near to where I am today without everybody. I've also had a lot of privileges, structural privileges in the world that have helped me to get where I am. And I want to acknowledge those privileges for helping me to get to this stage. So I definitely want to make it a story about me, but also a story about everyone else who's kind of helped me get where I am today. So what are the 4 ages? Jessica asked me just now, what are the 4 ages? These are the 4 ages of me. And we start with the golden age. I wouldn't pay too much heed to the colours. I'm just giving Ovid a little bit of a nod here, really. But the golden age of psychology, which is where I sort of started my academic career. The silver age of neuroimaging genetics. And for those of you who don't know what neuroimaging genetics is, I'm going to explain that shortly. The bronze age, which really should be the golden, well, I don't know if it should be the golden age, but is brain age. And as some of you have heard me talk about brain age till you're probably very sick of the concept, but some of you might not have heard of it before, so I'm going to give you some more details later. And then the final age is the current age, which I've called the manifold age. And some of you will know exactly what I mean by that, but I'll explain exactly what that means when I get there. So, to start with, we're going to go to the psychology age. And this is the age really where everything started. So I should have known that I was going to spend most of my life in universities because I actually spent 3 months living in a bungalow in the architectural delights of Loughborough University when I was age 7. We weren't in the high-rise tower. We were in one of these lovely concrete bunkers right in front of it. And my dad, he's here in the second row, he was head of personnel, or as we call it, human resources now, at Loughborough University at the time. And we were looking for somewhere to move to, and we ended up living there. And I really kind of got used to university life from an early age. And actually, I can credit Loughborough University, and my dad as well, for really sparking my interest in psychology, because he used to periodically bring home paper prospectuses from Loughborough University and kind of leave them in the vicinity of me and my brother, hoping that we'd pick them up and think, oh yes, we want to go to university. I couldn't find a picture of the 1998 prospectus, because turns out there aren't any pictures online of that. So this is the oldest Loughborough University prospectus that I could find. And so the 1998 one comes from a heady era when, we used to, a long time ago, used to have a Labour government, and we used to have England getting to the quarterfinals of major footballing tournaments, none of which, oh, hang on, yeah, anyway, so some things do go around. So I remember looking at this prospectus with great interest and coming across this page that said psychology. And I thought, hang on, what is psychology? And I read all about it, and I read all the course requirements, like, hang on, so some people, you get to study how and why people behave. I'd often questioned some of the behaviour of my peers, possibly including my brother, and thought, why are they behaving like this? What are they doing? And I realised that you can actually study that as an academic topic. So I decided, OK, psychology, that's the one for me. As part of my continuing love of spending time at universities, I did my work experience when I was in year 12. In fact, I did my year 11 work experience at Nottingham University, but my year in the materials engineering department. But my year 12 work experience was also at Nottingham University, this time in the School of Psychology. So I actually got to, get to see what it was like to be at a university studying psychology just for a week back when I was 16 years old. And I obviously really enjoyed it because I thought, yeah, this is definitely what I want to do. I was studying psychology at A-level as well and really enjoyed it. So I thought, right, I'm gonna do a degree in psychology. When I was trying to decide where to go to university, I considered UCL, I considered Warwick, I considered Birmingham, I considered lots of different universities. But I thought Bristol was actually where I was born, and it's a great city, it's got great academic credentials, really good scores, and I thought, I could see myself living there. No other possible reasons why I might want to go to Bristol. But it did mean I could go and watch a lot of football matches, so that was very nice. Whilst I was at Bristol, I had a great time, but it's here that I got my introduction to neuroimaging. So I was studying psychology, and I was learning about how people behave, and I was learning about the biology of behaviour and things like this. And then I had this lecture on neuroimaging, and it really spoke to me because I thought, actually, this is a way of not just studying behaviour, it's understanding what's inside people. Let's actually open the bonnet, see what's inside people, see what's in their heads. And that can really help us understand in a really scientific way why we behave in certain ways, why mental illnesses happen, why neurological illnesses happen. I thought this also gives me a good excuse to have a picture of my own brain in the talk, which is something I always like to do. So these are 3 different angles of my own brain. You can just about make out my nose in the axle here. But if you want a better view of my brain, I'm going to show you this. I need a volunteer. Jessica, I'm looking at you. Can you come here? So if anybody would like to feel my brain, Jessica will go round and if you can pass that round the audience so people can really get their hands on my brain, you're very welcome. I think she's had enough of my brain, haven't you? So yeah, feel free to pass that around. Jessica, if you can keep an eye on it. It's not that precious, but you know, maybe if it gets broken, I need an excuse to get a new one. So yeah, having spent my teenage years and my undergraduate years really thinking, I'm fascinated by psychology, I realised that actually it's not just psychology, but it's also neuroscience that I'm really keen to. But also, my undergraduate experiences not only introduced me to neuroimaging, but also to the study of abnormal psychology, mental health, and psychiatry. So it was after I finished my first degree, I decided to move on to try and do a higher degree. And it's this that I was lucky enough to be able to continue my career, where I moved from psychology to psychiatry. And this commenced the neuroimaging genetics age. So I was very fortunate to get a fully funded PhD place at somewhere with a lot of acronyms. So I did a PhD at the KCL IOP MRC SGDP Centre, which, to make life easier for short, we referred to as the SGDP Centre. SGDP stands for Social, Genetic and Developmental Psychiatry. And I spent 4 years there doing a master's initially, and then another 3 years doing a PhD at the SGDP Centre. My supervisors were Peter McGuffin, who at the time was the head of the centre, but also the dean of the Institute of Psychiatry, and Cindy Fu, who was both of them who was a psychiatrist, both of them psychiatrists, and they were a great help. Peter from the genetic side, Cindy from the neuroimaging side, for really helping me get to grips with some, what I found challenging times. And really helped shape me and my career and my attitudes to things. I'd also like to say that very sadly Peter passed away earlier this year, and it would have been great to have someone like him here at this event. He was a great mentor and a very kind person, and I know that I don't know if any of you guys here would have come across Peter, but it is sad that he wasn't here to see me do this talk. So yeah. But I had a great time at the SGDP Centre. I was able to publish my first ever academic paper, which I was very pleased with myself about. I was the first author. This was in 2008. Peter was the second author, and Anne Farmer, who was his wife and also a psychiatrist, it was the final author. So this was an editorial, and it was a kind of an updated commentary on kind of how depression is classified. And amazingly, we got it published in the British Journal of Psychiatry. And what was quite amusing was that they wanted to run it as an editorial, so they asked me for a photograph. This was back in 2008. I did have a digital camera. I definitely didn't have a smartphone in those days. It's like, well, where am I going to get a photo? This is actually cropped from a Facebook picture of me on a night out with my friends. Because it was the only photo I could find where I was actually looking at the camera, and it was actually a full face photo. So I don't know how many drinks I had by this point, but you could tell I look quite happy, and I thought that maybe that's a fitting way to start my publication career. So after that success, I, as part of my PhD, I had the good fortune to be invited by Cindy, actually. She suggested that I go and spend some time in Paul Thompson's lab at the University of California, Los Angeles. And as you can imagine, it was quite a tempting offer. LA is a very sunny place, and I made some great networks there, great collaborators, and I'm still in touch with quite a lot of the people I met there. The downside with going there is here I really got stuck into manual segmentation of the hippocampus. And I used this software that you can see in the top right-hand corner. It's called MultiTracer. And this was back in the days before we had automated segmentation of the hippocampus. So I spent many hours using various bits of kit like that to try and basically draw circles around very grainy brain pictures. I think overall I did something like 300 images, that's 600 hippocampi in the course of my PhD. And I vowed at the end of it I would never manually segment another hippocampus. And I have stuck to that vow. I've done other brain regions since then, but never again the hippocampus. But it did allow me to make what I thought at the time were very pretty pictures like this, and that's some hippocampal statistics from my thesis that I did there. It was also during my thesis that during my PhD, that I really got introduced to this idea of machine learning. When I started out doing neuroimaging, there was a lot of focus, and there still is, and quite rightly so, on what you might call mass univariate analysis. And you can see these brain images in the top here. Some people lovingly refer to that as blobology, because blob maps on a brain. And basically, this is a 3D rendering. And each of these tiny little cubes, which we call a voxel, is a little bit of information about the brain. And generally, the idea in these mass univariate analyses is to run a statistical analysis at each and every one, and then try and summarize them at the same time. But I remember going to a lecture by Mick Brammer, who was one of Cindy's kind of mentors, and he was talking about this alternative approach where you can actually do something which you might call pattern recognition, where you try and take into account all the different bits of information at once, and this was called machine learning. And at the time I thought, hang on, this makes loads of sense. And basically ever since then I sort of thought to myself, we should be doing more machine learning and less mass univariate analysis if we really want to get to a point where we're taking in the whole pattern for people to really understand. So yeah, I was able to complete my PhD in 2010. I defended my thesis whilst with a moustache, thank you, darling, which I had grown for Movember, so it was in November 2010. And I think Peter, my supervisor, thought the moustache was grown in homage of him because he had a very good moustache. But actually I was raising money for prostate cancer. So this is a photograph of me from 2011, actually, when I had my graduation. I'm wearing the same robes that you can see now. Not exactly the same robes, because these were rented, and so were they. The eagle-eyed among you might notice that I think I'm actually wearing the same shirt and trousers 14 years later, which says something about my appetite for shopping for clothes and things like that. But it's good that I still fit them, actually, so I can't really complain. So after that, I was able to get a postdoctoral role at King's, where I carried on doing research in major depressive disorder. And then I tried to apply for a few fellowships and wasn't successful, so ended up getting a position at UCL here, where I was working in the Huntington's disease group. And for me, that was quite a transition, because I was moving from having been immersed in a psychiatry world to something where the focus was very much on neurology. And I spent a couple of years at UCL in the Huntington's group, again, before moving on to Pastures New. And that Pastures coincides with the beginning of the third age that I want to talk to you about, which is the brain age. And this really was the transition for me from neurology to neuroscience. And it's probably when I first started calling myself a neuroscientist. So I was based at a great lab called the Computational, Clinical and Cognitive Neuroimaging Laboratory, or C3NL, and we were based in the Burlington Danes building over at the White City campus of Imperial College London, and I had a great experience at C3NL, and I made some really good friends here. And here's a nice photo of some of us. I think one of whom is in the room at one of our conferences, having some nice Japanese food in Geneva in 2016. But it was a really great lab, and there were some really great people there. And it really helped me grow as a researcher and as a person. And yeah, it was a really influential period in my career, being part of the C3NL kind of family. And it was while I was working at C3NL that I got involved in a project on HIV. So this project was called the COBRA project. It's not my favourite acronym because it stands for comorbidity in relation to AIDS, and they've really had to sort of shoehorn in the various letters there. But they did make a nice logo, so I kind of forgive them for that. And the idea behind COBRA was basically to look at this idea of accelerated biological aging in people who had HIV but was being successfully treated with antiretroviral therapies. So the whole idea was to try and look at measuring people in a biological way and see if it's affecting their aging process. And I thought this was a really interesting concept, and it's not something I'd really come across before. So I did a bit of reading around, and I came across this work where people were actually looking at biological age of the brain. So they were predicting age from a brain scan. And for me, it was this paper by Katja Franke and Christian Geyser, where they basically showed you can take a brain scan, do some stuff to it, and then get an accurate prediction of how old that person is, as you can see on this chart. That I suddenly thought, hang on, this could be something I could use for the COBRA project, because everyone there is looking at biological aging. Maybe I could look at brain aging. So that's what I then spent the next 10 years doing, basically. So for those of you who don't know, I thought I would explain a little bit about what brain aging is. I also wanted to include this image, this graphic, because it's the only time I've ever paid to use an image on the internet, paid for the royalties to use it. So I try to use it as often as I possibly can. I think it cost me about £3.99 about 8 years ago, but I want to get my money's worth. So it gives me an excuse to put this in here. So what is brain age? This figure I've trotted out a lot. It's quite useful, partly also like the colour scheme. But basically, the idea behind brain age is that we take a lot of healthy people, and we know what their chronological age is. They could be 18, they could be 50, they could be 90, and we have a brain scan from them. We do some processing to the images, and then we do some kind of statistical model, probably a machine learning regression, to predict how old those people are, to basically map from the brain scans to the age. We then do some kind of validation where you're kind of comparing their real chronological age with their brain age and checking that it's accurate. And then when you're happy with the accuracy of the model, you can then take the model, the coefficients, the weights that you've learned from the model, and then you can apply that to any new brain that you've got. So you don't need to know the age of the new brain, and the model will just tell you, oh, this brain is 61.7 years old. You can then take that age and then compare them to the real age. And if somebody's brain age appears older than their real age, it kind of suggests that their brain is aging less healthily than you might expect. And if their brain age appears younger than their real age, then perhaps they are more healthy. And obviously, you would expect in most healthy people, their brain age and their chronological age would essentially be the same. And then we can look at the difference between their brain age and their real age, just simply subtract them. And then you've got something which some people call the brain age gap, the brain age difference, the brain age delta, all those sorts of things. And I've been able to use that idea in lots of different contexts since. But the first two contexts for using this were in HIV as planned, but also in the context of traumatic brain injury. So at the time when I was at Imperial, I was working with David Sharp, who is a neurologist who leads a group who do research into TBI. And this was the first paper. And actually, for me, this was a really big deal because getting this accepted this is the first time that I've actually been able to do a kind of neuroimaging paper where machine learning is front and center behind the methods. And I can actually remember getting the acceptance email from the editor of the journal Annals of Neurology on Christmas Eve 2014. And I even remember where I was. I was actually at your house, Margaret, checking my emails probably when I shouldn't have been, and I should have been helping get ready for Christmas Day. But happily, they also decided to take one of the images we had from the paper and put it on the front cover of that. And I think that's the only time I've ever had a cover image was of that one. So for me, this felt like a big deal. And actually, in some ways, it has turned out to be a big deal because I've published lots of brain age papers since, and that was really the first one. And what we showed in this paper, and Rob is here, who's one of the other authors, is that people who've had a traumatic brain injury but recovered have an older appearing brain than you might expect. And the same is also the case in people who have HIV, despite it being treated. And we've used the brain age concept in lots of other contexts since then. Arguably, and I don't know who I'm arguing with, maybe I argue with myself, but arguably my favourite brain age paper is this one. And this was using a data set from Scotland called the Lothian Birth Cohort of 1936. And I give a lot of thanks to Ian Deary and his colleagues for letting me use this amazing data set. So this is a study of people who were all born in the same year, and they did an IQ test when they were 11 years old, so in 1947. And then in the early 2000s, these people were recontacted and enrolled in a genetic, behavior, biomedical history, and neuroimaging study. So they all had a brain scan at the age of 73. So what we were able to do is take their brain scans, compare them to that model that I showed you on the previous slide, and then see whether their brain said how old they were. And remember, we hadn't actually shown this model the real age of these people at all. And when I ran the analysis, I was delighted to see that actually it sort of worked. And basically what you can see is this is age on the bottom here, and then on the side is how old the model thinks they are, their brain age. The green points are all the training data set. So we've got people across the lifespan. But in the red points, we've got this very narrow band, because that's the real age of the Lothian birth cohort, who were all born in the same year. But then the average is actually 73. So it was sort of correct, which is very reassuring. But what we were really interested in, which is on the second plot, is the variability in brain age. And what does that variability in brain age actually tell us about people? And actually it tells us that they're not aging very well. So these are all different ways of measuring aging that the guys in Edinburgh had already measured. So G is a kind of cognitive measure of how well your cognitive aging is going. Grip strength is a measure of musculoskeletal health. Then FEV is the forced expiratory volume, which is lung function essentially. And then 6-minute walk time is a measure of your gait speed. And it showed that people who had an older appearing brain, according to their MRI scan, also had poorer cognition, weaker grip strength, poorer lung function, a slower walking time, and also a higher allostatic load. And allostatic load is kind of a composite measure of lots of blood tests and physical measures and BMI and that sort of thing that also goes up with age. So for me, this was a really strong validation that what we're measuring with these brain scans is actually related to something that relates to aging that you can measure in different ways. And I was very relieved to see this, because it really kind of validated this whole idea of, is it aging? Is it telling us something useful? To go further than that, this is a slightly morbid slide, but it's still very interesting scientifically. So as part of the study, the people who run it knew when the people in the study had died. And we were able to look and see whether their brain age at age 73 affected whether they had died by the time they were 80. And essentially what you can see is that obviously, so about 10% of them had died at the time. And although it's slightly different between males and females, in both males and females separately, those who had died after their brain scan had older appearing brains at age 73. And if we stratify them by people with an older appearing brain and people by a younger appearing brain, we can show that they were more likely to have died if they had an older appearing brain at the age of 73. And the fact that brain age is relating to mortality in this way, I think is a really interesting aspect of how measuring the brain can tell us something about the whole of the body's health. Because it's not like these people were dying of brain diseases, they were actually dying of cardiovascular disease. But clearly the brain is connected to the rest of the body, and if the rest of your body is not very healthy, that's going to be reflected in your brain health. So this was really a really nice validation of the whole kind of idea of it. And I think it really helped propel brain age as an idea into kind of the mainstream, and people talked about it. And actually I even got a bit of exposure in the media. I should have probably found this actually, but I did a few interviews with people, including this Mexican TV channel that came to Burlington Danes building and interviewed me. I wanted to crack out some of my Spanish, but actually I'm not very good, so I did it in English. And I even did a little bit for Sky News as well, where they sent one of their reporters to Imperial, and we put them in a scanner and we calculated how old their brain was, and it went out on the evening news. So it was quite good fun. I've never had any kind of exposure like that since then, but it was nice to happen once. So another paper I wanted to mention is this one, and this is actually my most highly cited paper, and this was a collaboration with Giovanni Montana and people who were then at King's while I was at Imperial. It's a bit confusing. But this paper was the first application of convolutional neural networks, or deep learning, to the prediction of brain age. And we started this work in kind of 2016. And at the time, deep learning seemed really cool and really new and really fresh. And it was actually quite exciting to do this. I didn't at the time know that it was going to go on to be really highly cited, but it just showed you that we kind of got there at the beginning of a wave. Obviously, other people have been doing deep learning for a few years before this and done way more stuff on deep learning, but the specific combination of brain age and deep learning seemed to be very timely. The other great thing about it is because it's deep learning, I was able to say, I do AI. Fast forward 7 years, it's really useful to be able to say, I do AI, I do artificial intelligence research, because that's what funders want to hear, that's what newspapers want to talk about. So, you know, it is AI, and I won't hear any arguments against that. So after my time at Imperial, I was very lucky, and I'll credit Rob Leach for this, because he's the one who told me about this. I applied for a fellowship for the UKRI, which I was able to get, and you know, I was able to move back to King's College London, so back to Denmark Hill. It wasn't that I was just missing the architecture, but I was able to move this time to this neuroimaging department at King's College London. But I wanted to point out that, so this was a successful fellowship for me, but this was the 11th application for a fellowship that I'd done in my career to this point. The previous 10 had all been unsuccessful, and yeah, I mean, Danny kindly said at the end, I'm quite persistent, and at the beginning, sorry, and I think I am. I think I am quite persistent, and you know, despite having lots of knockbacks and failures, I think you know I was very well supported with people, and people kept believing in me and supporting me, and eventually, after 11 applications, I got this fellowship, and I wanted to take a little moment to talk about this because I think it's really important to normalise failure. Because in academic careers, everybody will fail. And in some ways, the most successful people aren't the smartest or the most brilliant, but they're the most persistent or the people with the thickest skin. And I think it's, you know, we all will fail at things. And I think it's really important that we talk about our failures as well as our successes. And I wanted to use this as an opportunity to talk about all my failures. I won't give you the blow-by-blow of all the 10 failed applications, but I did learn from them, and eventually it all paid off. So I'm happy. While I was at King's, I produced my software package, BrainAger, which some of you have used. I think it's been downloaded at least 7 times, so you know, really happy with the impact that that's made on the field. And I still get people emailing me now, even though it's 5 years later, saying, oh, this thing doesn't work, or why is this giving me that result, and that kind of thing. And I think, I'm really glad I put that out there. But in fact, where's Rohan? We were even bemoaning it giving weird results today, weren't we? So it's still limping on. I was also able to set up this time the Enigma Brain Age Working Group. So Enigma is actually a really big consortium set up by Paul Thompson, and it's a really good acronym. Enigma stands for Enhancing Neuroimaging Genetics Through Meta-Analysis, which I really think follows the strict rules which Neil Oxtoby lays down for doing a good acronym. And as part of that initiative, there's lots of different working groups, and we were able to set up a Brain Age working group where it's basically a collaboration of about 90 or 100 researchers from around the world who are all interested in brain age as an idea. We have periodic meetings and a kind of mailing list where we share some of our papers and some of our results. And it's been a really great, for me, it's been a really great thing to be involved with because there's just such goodwill and positivity amongst this consortium. Nobody's really in it for the glory. Nobody's really in it because they think, oh, they're going to, you know, take over the world. It's actually a really great way to kind of interact with people from all over the place who've all got a common interest. And I also like it because it means I get invited to the Enigma drinks night at OHBM every year as well. So that's another fringe benefit. One of the other things I'm quite proud of from my time at King's is doing some neuroimaging in the real world. And Fran has been very involved in this study. In fact, she's the lead author on this paper. And this was work done as part of a memory clinic at the IOP in Denmark Hill, where we had people coming in for scans over a 10 or 12-year period from all these different boroughs in South London, which, as you probably know, is a very diverse, ethnically, but also socially kind of diverse place. Lots of neuroimaging data, but lots of data sets generally in the world are of kind of very narrow demographics of kind of healthy volunteers from well-educated backgrounds who've got the spare time to take part in research and are predominantly white and not very representative of the real world. So it was great to actually get some real-world data. So all of these people who came in had been experiencing some kind of cognitive symptoms, and they'd gone to a memory clinic. And they all have an increased risk of dementia, although they obviously won't all get dementia. And what was great about this study is that all their brain imaging data was linked to some electronic health records. So we could actually link it up and see what was happening and see what happened to those people. So this was the barcode study, and this was a Wellcome Trust Seed Award, which I got in 2018. And for me, that was also a big moment because that was the first kind of independent grant that I'd got, and that really helped build my confidence and think, well, actually, I can make a career out of this. I haven't included the results of the study here. If you want to know more, you can grab Fran at the drinks later. I'm sure she'll tell you about it. But the spoiler is that actually brain age does help predict whether people go on to get dementia or not. And the final thing to say about my time at King's was also getting involved in the MIDI study. MIDI, this is not one of my favourite acronyms. It stands for Medical Imaging Abnormality Deep Learning Identification, which really breaks a lot of the rules, but we're calling it the MIDI study. And this was really using deep learning, for, again, very much for clinical, real-world neuroimaging. And the idea behind the MIDI study is basically to take hospital data, so every brain scan that had ever been collected, basically, at King's College Hospital, and then all the radiology reports that go with it, and then try and learn how to figure out automatically whether a brain scan has an abnormality in it or not. And this study's been led by Tom Booth with some great work by David Wood, who are both still involved in the project, and this has been a great thing for me to be involved with because it's really allowed me to kind of get to grips with big data, real world data, and to kind of expand beyond just doing brain age stuff. And the MIDI project's still going strong today. So that was all started when I was at King's. But whilst I was at King's, I saw a job advertised at CMIC. And I thought, well, that looks like a great job. I'd love to work there. I know some of the people there are really great. And I'd love to do it. So I had a go and applied. And luckily enough, I didn't get eaten alive in the interview of 7 professors all staring at me. And I was able to move from King's to UCL. And that commenced the final age, which is the manifold age, which is really where I kind of transitioned or transformed from neuroscience to computer science. So as Danny mentioned at the beginning, my role really is to try and, I'm based at CMIC in 90 High Holborn in the nice shiny building you can see there, but it's also to try and work as closely as possible with the Dementia Research Centre, who are based in Queen Square. And for me, it's been a great privilege to kind of sit between these two different worlds, one of computer science, one of sort of clinical neurology, because I think they're so complementary in so many ways, but often people in those worlds don't really speak the same language in terms of the research that they do. And for me to be able to kind of be a bit of a conduit for that and to encourage other people to kind of be involved in both of those things, I found really inspiring. And I think we've made some really good progress towards kind of taking computational methods into the clinic for dementia. So this is a slide, actually, that I found from my job interview. So you can ask Danny as to whether I've actually done what I said I was going to do. But basically, a lot of the ideas behind the research that we've sort of been doing with the DRC and CMIC is to take imaging data, whether it's MRI or PET, somehow store it alongside other data sets, and then make a clinical prediction based on what we think their brain and their other biomarkers are telling us about their risk of progression. And that's the kind of idea that we've been pushing forward. And to support that, me and my colleagues decided to form the MANIFOLD Lab. And there'll be a quiz at the end to see if you can remember what it stands for. No, I'm joking. But yeah, so this is the Machine Learning, Artificial Intelligence, Neuroimaging, Focusing on Longevity and Dementia Lab, or the MANIFOLD Lab. There's a good representation of people from the MANIFOLD Lab here, so it's great to see you all here. We spent quite a long time agonising over the name. One of the options was Gandalf, which I think in retrospect I'm glad we didn't go for, as much as I love Lord of the Rings. Yeah, I'm very happy with that. And thanks to Melis, who's an alumnus of the lab, who designed this lovely logo. So this is the lab, and to be honest, being part of this lab is probably my proudest academic achievement, because I think all the guys in the lab are really amazing, and I've loved working with you all, and I look forward to working with many of you, for a long time into the future. So here is our Christmas party. You can see people like Tragstar in the background, and this was a murder mystery night at a pub in King's Cross, which we enjoyed. And here's a more recent photo from some of us, Manifold plus a few friends. standing outside Psy's Gangnam Style hands outside the conference centre. We were just a couple of weeks ago in South Korea. And I think one of the great things for me about the lab is that not only do we do great research together and everyone's very collaborative, but we also have a really good time and enjoy each other's company. And for me, that's a really important part of being a researcher, being a scientist, being an academic, is actually being able to spend your working life with people you like, and I hope that continues. So what do we do in the MANIFOLD Lab? Not just have fun, but we do research into Alzheimer's disease, we do research into Lewy body dementias, we do research into multiple sclerosis, motor neuron disease, autoimmune diseases, neurodevelopment, and aging. So we've really got quite a broad range of different areas that we look at, and it's really expanded. And I think part of me sort of chopping and changing between diseases over my career has really led me to have quite a broad view of how different methods can be applied in different places. And it's not just brain age we do, it's beyond brain age. We do things like normative modeling, we do explainable AI, we do data fusion, we do latent space generative modeling, we do automated machine learning, image segmentation. We're working now on foundation models and joint text and vision modeling. And I think these are all really exciting data analysis techniques, and I think they're really going places, and already we've made quite a lot of impact in some of the applications, and I think there's a lot more to come. So although the brain age was a great age for me, I'm glad it's not just the brain age anymore, and I hope the future is going beyond brain age. As Danny already mentioned, one of the things of the future is this lovely machine, which we still haven't given a name for. We need to think of a good name. So I'll take suggestions as to what you should call this. This is the Hyperfine portable MRI scanner, which we were able to purchase earlier this year. And these are some of the scans you can get from this. Important to note is that the field strength, the magnet behind this, is about 30 times weaker than the sort of scans that we normally use. But you can still get very clear pictures of people's brains. And the great thing about them is they're portable. You can plug them into the wall. And they're much cheaper and easier to operate, and they're much safer, they're much quieter, they're much less claustrophobic. And for me, I really think they can play an important role in taking neuroimaging away from the hospital, away from the research centre, and into the community, and into resource-scarce settings. And I think these and other low-field systems that are going to come out over the next few years could really change how and where people get scanned, and really kind of democratise brain imaging, which for someone who loves brain imaging, that's great. And it will leave its ivory tower a bit and go more to the people. I've nearly finished, bear with me. So there are some challenges that remain for neuroimaging, and obviously I'm not going to solve all of these challenges, but it's clear from lots of research that the burden of brain diseases is still going up globally. We don't have great treatments for a lot of things. The clinical demand for neuroimaging is increasing. More and more people want to have brain scans as part of their clinical practice. But often it's too expensive and inaccessible, and there aren't enough radiologists to cope with the demand. Even if we, the demand for people to report the scans far outstrips the capacity for doing it, and we can't train enough radiologists quickly enough. It's not going to help. And a lot of we do have predictive models that can take brain imaging data and tell us something about people, but often these models are inaccurate and biased, often by demographics. So I hope in the future we will be able to transform the way this works, and we'll be able to automate some of the aspects of neuroradiological reporting, so that we don't need, we don't have this mismatch between the number of radiologists and the amount of work they have to do. I hope that we can add quantitative neuroimaging metrics to clinical decision-making processes to really make these models more accurate and more informed. These models need to be trustworthy, they need to be unbiased, they need to work for people irrespective of your demographics. And we really need to work to integrate these markers, these brain imaging markers, with other biomarkers, be they from blood, cerebrospinal fluids, genetics, all these sorts of things. And I've believed this for a long time, and I still believe it, but I really do think that data analysis will be able to help patients with brain diseases. And in some ways, that's been the undercurrent for me of all the stuff I've worked on over the years, is that I want to help people with brain diseases, and I think analysing brain images is the way forward. So, in summary, these are my 4 ages: the psychology age, the neuroimaging genetics age, and the brain age. And then the manifold age, which is the current one. And in some ways, it has been a bit of a metamorphosis for me. It's been a story of transformations from psychology, psychiatry, neurology, neuroscience, computer science. I've left the bottom one blank because I can't say what's happening next. I might transform into something else, I'm not sure. But at the beginning, I promised I'd also this would be a story of gratitude. So I'm going to move on to the soppy bit now. So first of all, I want to thank the people involved in the slide. When I started writing these slides, I started putting everyone's names on it. I realized this is just going to be too many names, and most people aren't going to know who they are. And then I was worried about missing people out, and I just, had a bit of a crisis about it. So I thought, actually, I'm going to go high level. So my psychology age was brilliant, and I was very happy. I made some great friends from my hall of residence in Bristol, the Wills Hall boys, who I still go on holiday with every year now. I made some great friends from my psychology course, who I still, see very regularly. And in the background as well are my school friends, who I've, this is on the right-hand side is a postcode map of NG2, which is the postcode that I grew up in. And the NG2 crew is still going strong, even though we haven't been in the same school for 25 years. So yeah, thanks to all those guys for really supporting me and believing in me and just generally being great friends. I want to thank people at the SGDP. There's Peter McGuffin and Cindy Fu, who I mentioned earlier, as my supervisors. But also I made some great friends at the SGDP Centre. People like Tori and Soph and Gemma, Nick and Andy, and all those guys. And I really loved being a PhD student. And I hope that PhD students in CMIC can have as good experience as I did when I was there. I also had loads of great colleagues and collaborators at the IOP. So it's, I want to acknowledge them, because it was a very collaborative experience for me. The third age of thanks is brain age. It's great that Rob is here. I think he's probably the only person from C3NL who made it here today. But I also had a brilliant time there. And that's where I really felt like I actually had my kind of first intellectual academic freedom to really do what I wanted scientifically. And I think that's really set me in a good stead for the rest of my career. Partly that was helped with the great collaboration with Amsterdam to really do that HIV project, to really start off brain age for me. Part of it was thanks to the guys in Edinburgh who helped me use the Lothian birth cohort to actually do that study I talked about. But also the people at King's, when I went back there, were really welcoming. And in fact, it was so good, Rob even followed me there and is still there today. And I go and visit him sometimes as well. So thanks to everyone who helped during the brain age. During the MANIFOLD age, I want to thank the MANIFOLD Lab. You guys are amazing. I love you all. And I want to thank everyone at CMIC as well. Danny, thank you very much for pushing me to go for promotion and for pushing me to do this talk. I've really enjoyed it. There's lots of other people at CMIC who've been great and have made me feel really welcome. There's also people at the DRC that I collaborate with, and I really appreciate their input, and I enjoy working with them. But also more widely at UCL, working with people at Buckney, in the MS Centre, and also other bits of the Institute of Neurology, and loads of collaborators all over the world now. I feel really privileged to collaborate with people from so many different countries. It really makes me feel how international science can be and how we should have no boundaries and borders, and that we can all work together and achieve great things. My final thanks are my thanks for all ages. And I want to thank my family, particularly my parents and my brother, who've always supported me. And even when I pick weird combinations of A-levels and decide to have gap years going around Mongolia and stuff like that, they still supported me and believed in me, and I really appreciate that. I want to thank my children, who are in the picture as well as in here in person. It's been a real privilege being your father, and I look forward to being your father for the rest of my days. Even when you end up in hospital and I have to sleep in a fold-out bed next to you a few nights before my lecture. And I'd just like to thank Sarah, my wife, who has been a constant support, even when I've doubted myself, which happens quite a lot, and inspired me with everything that she does, and is a great parent to my children. And thank you so much. And thank you all for listening.